I have the honor actually introducing a friend, a colleague for over 20 years, a mentor, and the keynote speaker, Dr. Stanley Naides. He is a board-certified internist, especially certified in rheumatology, and this is how we met at Penn State University, the College of Medicine at the Rheumatology Department as a postdoc. I have to say that Stan has immense experience in big data, setting up biobanks, venturing in helping patients and clinicians alike all over the world. His career, yes, was academic. He left me to go to Quest Diagnostics, and he did great things with Quest. And then he moved to Euroimmun, and also now he settles in Labcorp. But as a laboratorian in a commercial high complexity test reference lab, his research portfolio includes test development, immunology, autoimmunity, rheumatology, obviously. He has served on NIH review and foundation grant review study sections. He continues to serve on national professional societies, committees, and is currently the chair and board of directors of the Autoimmunity Encephalitis Alliance. Dr. Naides is the author editor of more than 100 peer-reviewed papers, books, chapters, reviews, and still going strong. And we really have the honor that he's here in person. So thank you.
It's a great honor to return to Jyväskylä and an honor to be here and a pleasure. And I'm going to tell you today about a story that begins in Iowa during an outbreak of parvovirus. And as a rheumatologist, I had the opportunity to see a number of patients and ask some very important questions along the way. By way of disclosure, I am an employee of Labcorp, which is a reference laboratory in the United States with an international footprint. So what I hope to accomplish in the next 45 minutes is to describe the epidemiology and clinical manifestations of acute parvovirus B19V infection, at least briefly, and to describe the mechanisms by which B19V will induce apoptosis or programmed cell death. And you'll hear me refer to the virus as B19 because that's the older nomenclature I grew up with, and they added a V for virus after it more recently. And ultimately, I'd like to describe the mechanisms by which a virus can break tolerance to self-DNA, causing autoimmunity. So the question is, can an acute viral infection lead to autoimmunity? And perhaps the next question is, why does native DNA elicit autoantibodies, the hallmark of autoimmune diseases such as systemic lupus erythematosus? Because we know that mammalian DNA is a poor immunogen, and that immunization of mice with human DNA does not elicit any anti-DNA antibody response. But yet in systemic lupus erythematosus, we see characterize this disease by autoantibodies to native double-stranded DNA. So how does that happen? Well, the story begins back in 1986 during a major outbreak of parvovirus infection in southeastern Iowa, which brought my attention to this particular virus. And to level set our discussion, B19 is a single-stranded DNA genome. It's non-enveloped. It's extremely small, hence the term parvo, or the Greek meaning small. It's a small virus. And the major proteins that are produced are two structural proteins, VP1 and VP2, as you can see on the example on the bottom right. This is a single-stranded DNA, has a single upstream strong promoter, a non-structural protein which functions as a helicase, and two viral capsid proteins which are in the same open reading frame, but with alternate start sites. VP1 is the minor capsid protein that sits at the vertices of the five-fold apex of the icosahedron, and VP2 is the major capsid protein, and VP2 by itself can form empty capsids. And so what we do have minor proteins that are produced of unknown function. There's a 7.511 kilodalton and more recently a 14 kilodalton protein. But because this virus is very simple in its structure, it's dependent on host cell DNA replication machinery and cell cycle, and it's species and tissue specific. And as a clinician, I was often asked by pet owners, could I have gotten my parvovirus infection from my dog, because canine parvovirus is a known entity for which dogs are vaccinated? And the answer is no, these are species specific. There are several genotypes worldwide, B19, and actually after COVID in 2023 to 2024, there was a resurgence of B19 in Europe. And so people have been reporting a number of cases and various manifestations in Europe more recently. And there are a couple of other human parvoviruses that have been identified with a little bit of information about any potential pathogenicity. So what is the epidemiology of B19V? It's common. Half of us in the audience have seen this virus, particularly during childhood, and carry IgG antibodies. Outbreaks occur in late winter and spring, but also have been seen in the summer and fall, and they're always sporadic cases. The individuals that are at highest risk are those in occupations with frequent exposure to children, because typically as children come up to school, they get infected. And then every 3 to 5 years, as a new cohort of uninfected susceptible children arrive at school, we see more outbreaks. So the typical incubation period is 7 to 18 days. And then there's a viremic phase in which B19 DNA can be found in the serum. It's actually a virus. There's a viremia. And during this phase, there is a flu-like illness with malaise, although it can also be asymptomatic. And then after about day 11, as antibodies begin to appear, first IgM and then IgG, particularly to VP2, and soon after to VP1, which is required for neutralization, the respiratory virus is no longer shed in the nasal secretions, but that corresponds with the onset of rash, arthritis, and myalgias. So it's a biphasic illness, a viremic phase with flu-like illness, and then an antibody phase, which is associated with rash and arthralgia. The rash is caused in childhood, causes the classic slap cheek rash of fifth disease. In adults, the rash may be very subtle, and the arthralgia is a polyarthritis and can mimic rheumatoid arthritis in its presentation. So where does B19 like to grow? It likes to grow in erythroid precursors and shuts down production of new red cells. And you'll see that's one of the main causes of one of its hematologic manifestations, as we'll point out shortly. But the virus likes to assemble in the nucleus, and on examination of bone marrow, you can see in erythroid precursors an accumulation of virions within the cell nucleus. It tends to accumulate around the inner surface of the nuclear membrane. So there have been a number of major manifestations of parvovirus B19 infection. One is asymptomatic in individuals. And the next major manifestation that was identified was transient aplastic crisis. And so these are individuals with chronic hemolytic anemia, such as sickle cell disease, in which individuals require a brisk reticulocytosis to keep up with their rapid turnover of mature red cells. And so they have shortened red cell survival. And so if they stop making red cells, as they do with a B19 infection, their hemoglobin drops precipitously, and they typically require transfusion support until they develop antibodies and recover. The next manifestation that was identified was erythema infectiosum, the classic fifth disease rash illness of children associated with slap cheeks and a macular reticular eruption on the torso and the extremities. And then finally, in women who are pregnant, fetuses, when they're infected, it can be passed through the placenta. Fetuses are a little bit like a child with sickle cell anemia. They have shortened red cell survival. And so when they stop making new red cells, their heart has to work overtime to keep up with oxygen demand, and they develop fetal hydrops, essentially congestive heart failure. And we've also demonstrated that fetuses may also develop myocarditis, a viral myocarditis. And then finally, arthropathy in individuals who present with sudden onset of polyarthralgia, polyarthritis. We can see bone marrow suppression, particularly in individuals who are immunocompromised, whether they have HIV, have had prior chemotherapy for cancer, or who have a primary immunodeficiency. And finally, we can see isolated bone marrow lineages being suppressed, and we can see hepatitis. Less common have been a number of manifestations that have been defined, as you can see: vesicular pustular eruption, Henoch-Schönlein purpura, TTP, gloves and sock syndrome, a very unusual rash presentation in B19 patients who develop red hands and red gloves as if they're wearing red hands and red feet as if they're wearing gloves and socks. And acute fulminant liver failure, which we'll come back to and talk to more extensively. Occasionally, we've seen myocarditis, hemophagocytic syndrome, excuse me. Polyarteritis nodosa, granulomatosis with polyangiitis, paresthesias, aseptic meningitis, and a few cases of sensory neural hearing loss. So B19 enters the cell through interaction with globoside, a neutral glycosphingolipid, which is plentiful on red cell precursors and can be also seen on other tissues. But B19 will replicate in erythroid precursors. It will get into other tissues, but it tends not to replicate because it doesn't have the appropriate machinery to produce all of its transcripts, as we'll see. So the research focused initially on B19 in synoviocytes. And indeed, if you put B19 on normal synoviocytes, they develop a more invasive phenotype, much like synoviocytes in rheumatoid arthritis. But around 1992, I got a call from Alan Lagnus, who was a transplant surgeon at the University of Nebraska. I was still in Iowa. And he asked me, I have patients who have non-A, non-B, non-C acute fulminant liver failure associated with aplastic anemia. So I went to the literature, he said, and I wanted to see what causes aplastic anemia, and B19 came up. So I asked the rheumatologist here, what do they know about B19? And they said, call Stan in Iowa. So he said, would you test some of my livers? And I said, okay, let's do it right. Send me all your livers, send me controls and blind me, and let me see if I can identify the ones that are associated with acute fulminant liver failure. What was interesting about these patients is that when you looked at the pathology of the livers were not inflamed. What they had was massive hepatocellular dropout. It looked like the livers were dying through apoptosis, programmed cell death of hepatocytes. And so the question was, what would be the association of B19? Could we find B19 in these livers? The other interesting observation is that none of these patients had IgM antibody to B19, but they all had IgG, suggesting that if they were indeed infected, it was a past infection, not a recent infection. So we asked the question, can B19 induce programmed cell death in primary hepatocytes? And so what we did was to take viremic serum from B19. And at the time, one could not, still is very difficult to grow B19 in culture. And so fortunately, I was able to reach out to some of my colleagues, and this is pre-HIPAA, was able to reach out to colleagues and ask if they could send me serum from individuals who had a transient aplastic crisis. I did my internship in a large county hospital in Miami, which had a large African-American population and any number of patients with sickle cell disease who would show up in aplastic crisis in the emergency room. And so I received 5 sera, one of which was very early in the course without any antibody and high titer viremia, 10 to the 15th particles per mil, which is very typical of an acute B19 infection. And so that was initially my source for virus. And so if you take B19 and put it on primary hepatocytes and come back 48 hours later and ask if cells were undergoing apoptosis, yes, indeed they were. And the way we demonstrated that is by this observation. Phosphatidylserine is part of the plasma membrane of the cells, but it's asymmetrically maintained on the inner surface leaflet. But during apoptosis, that active process stops, and phosphatidylserine will be expressed on the outer membrane leaflet. And annexin, which is a protein that aids scavenging of apoptotic bodies and cells undergoing apoptosis, will bind to the phosphatidylserine. And typically, there are receptors on macrophages for annexin that will now gobble up that debris. And so what we use is annexin that has been labeled with a fluorescein tag. And indeed, you can see on this one, a number of apoptotic cells that express, that are determined to be apoptotic because they express phosphatidylserine on their surface and are labeled by annexin 5. If you irradiate the B19, you can see that you decrease the amount of apoptosis. So you need active, viable virus. And so we then ask what genes are expressed in these cells, and both in primary hepatocytes and HepG2 cells as a choriocarcinoma, hepatic cancer cell line. You can see that the non-structural protein is expressed. And here we take the cell lysates and we treat them with RNase to remove any RNA. We treated them with DNase to remove any DNA, and you can show that the RNA is remaining, and there's RNA both in the HepG2 cells and in the primary hepatocytes, but there's very little or no transcript for VP1 and VP2. So this is a restricted infection. And so you're getting production of the non-structural protein, but not viral capsid. So you can't make whole virus, or at least not very efficiently. If you get a little bit of VP1, VP2, you may make some virus. So we also then took out the gene of NS1 and put it into a enhanced green fluorescent protein expression vector under the control of an insect promoter. And the purpose of this was to simply say, we're going to put this into cells and nothing would be produced until we expose the cell to an insect steroid, which will now then make the NS1, which is tagged with the enhanced green fluorescent protein. It's a mechanism, a method by which we can actually see when NS1 is being produced. And indeed, as you can see, not only was the NS1 protein produced, but it localized to the nucleus because NS1 has nuclear localization signals on the protein, which allow it to enter the nucleus where virus is assembled. And so in summary, at this point, we knew now that B19 causes a restricted infection with production of NS1, but very little capsid transcript, and that it entered the nucleus, NS1, and that the apoptosis peaked at 72 hours based on annexin 5 staining It was inhibited by anti-B19V neutralizing antibody. It was inhibited by UV irradiation and was infectious dose dependent. So what would be the mechanism by which NS1 could cause apoptosis? So there are two major pathways through which apoptosis can occur. One is an extrinsic pathway in which a member, for example, of tumor necrosis factor activates the receptor, sends downstream signals that activates caspase 8, which in turn is an enzyme that will lead to activation of caspase 3, and further down, it'll induce apoptosis. Or internal signals, such as mitochondrial stress signals that activate caspase 9. And you could look at these different pathways using specific inhibitors to see what you could block. And so we did this, and lo and behold, the NS1 expression induced apoptosis required caspase 3, which is the branch point for both, but more important telling, it required caspase 9, but not caspase 8. And you can see the amount of apoptosis was inhibited by inhibitors of caspase 9, but not caspase 8. So there looked like there'd be then mitochondrial instability that was confirmed, as you can see in these experiments, the baseline apoptosis, and then caspase 3, caspase 9 inhibitors would decrease apoptosis, but not caspase 5 and some of the others. So what triggers caspase 9? We think it's associated typically with mitochondrial instability. And so one of the questions we had was, could DNA damage lead to intrinsic pathway activation? Because we knew a little bit about what the virus does, what NS1 does during a natural infection. NS1 is a member of the superfamily 3 of helicases found in small DNA viruses. It contains DNA binding, nucleotide binding, helicase activities, and nuclear localization signals. And it enters the nucleus as a dimer, and it functions as a hexamer. It's been very difficult to define the polymeric structure of NS1 because no one has been able to produce full-length NS1. But more recently, a number of Chinese investigators have looked at larger pieces of NS1 and have pieced together an understanding that indeed it forms a hexamer around DNA and performs, the DNA moves through the hexamer, and it actually may form a dodecamer, 12 units in some instances. And what this virus does, what this non-structural protein does, is it nicks and unwinds DNA. So B19 is single-stranded, but it has at either end a palindromic structure. So it forms a hairpin at either end where the DNA loops back on itself. And so when it extends from its 3' end during replication, it extends down. And so you have 2 copies of the genome, viral genome, which are covalently linked. And it gets to the end, and the 3' end, which is where it elongates, folds back on itself and copies again. So you get monomeric forms of a replicative structure, which has 2 copies, and then a dimeric form, which has 4 copies of the genome. And this virus will encapsulate both negative sense and positive sense copies of its genome. And so the non-structural protein is designed to nick those replicative forms, to reduce them to single copies that are packageable. And there was always a hint that NS1 seemed to be incorporated into the capsid. So there was thinking that perhaps it interacts with the DNA in such a way as to form a scaffold around which the capsid would assemble. And so we also know that NS1 is cytotoxic to transfected cells. apoptosis, and that it's a transactivator of IL-6, TNF-alpha, and it turns out more recently people have identified a whole host of pro-inflammatory pathways can be activated by NS1. So we asked the question, is NS1 a host cell DNA damaging agent? Will it do to host cell DNA what it does to its viral DNA target? And so we asked, will it cause single strand breaks, double strand breaks, or bulky adducts? Will it covalently link to the DNA? And so we looked at this by first asking, can we see activation of poly-ADP ribose polymerase, or PARP? And so what PARP does, when there is a single strand break in DNA, it attaches to the single strand break and it forms polyribosal strands, a polymer of ADP ribose on itself and any surrounding protein. Now, this is energy depleting. It requires a lot of NADH. It leads to mitochondrial membrane impermeability and release of apoptosis-inducing factor, and ultimately caspase-9 activation. So we asked ourselves, is PARP activated? And indeed, what we did was to express NS1 GFP protein in HepG2 cells and demonstrate that if you take a cell lysate, do a Western blot, you can show that 100 kilodaltons, which is what the NS1 GFP protein molecular weight is, that protein is poly-ADP ribosylated. And so indeed, we probed the Western blot with antibody to poly-ADP ribose, and indeed it is poly-ADP ribosylated. We could inhibit excuse me. We could inhibit PARP activation with an inhibitor and in a dose-dependent fashion and reduce the amount of apoptosis by about 50%. And so indeed, PARP was activated, and yes, there are single-strand breaks of DNA. We then asked, are there double-strand breaks? And we did this by a common assay in which you take individual cells, spread them out into an agarose gel, put them into an electrical field. And if the DNA has been fragmented, instead of remaining a dot, as you see in the normal, it would tend to blur out like a comet. Now, you may not see it very well in this projection, but it's there. And so what we found early on, within the first 24 hours, there were no comets in neutral conditions. And so in neutral conditions, even if you had a break, you had overlapping strands, you would not get separation. If there were a double-stranded break in neutral conditions, you would. And of course, in alkaline conditions, when you separate the strands, you would see a comet. There was no effect on histone 2A-X phosphorylation, which is seen in double-stranded breaks. And so at least early on, you're not seeing double-stranded breaks. At 48 hours, you do see double-stranded breaks, but that's likely due to the process of apoptosis when there's a burst of endonucleases released that chew up DNA. So the answer is no, not directly. We don't see double-stranded breaks. And then we asked ourselves, do we see bulky adduct formation? And what happens in bulky adduct formation is when a protein, for example, will attach to the DNA, it separates the strands locally. And it distorts the DNA, and it leads to an area of strandedness, which is recognized by a replication protein A, which binds to single-stranded areas. And that in turn allows this large protein, ataxia-telangiectasia mutated and RAD3-related protein ATR, to become activated. This ATR circulates, it has an inhibitor protein, which when the inhibitor protein binds to RPA, it phosphorylates ATR, And when ATR becomes phosphorylated, it activates P53. And P53 it leads to when it's phosphorylated, leads to mitochondrial potential changes, caspase 9 activation, and apoptosis. So another energy depleting process. And you can inhibit that with caffeine, which is a specific inhibitor of ATR and also ATM pathways. And so again, you could reduce apoptosis from about 35% of cells down to about half of what you would otherwise get without inhibition. And so it looks like we indeed form bulky adducts. And so the answer is yes. We further looked at this process by taking radio-labeled cells. We labeled the DNA with P32 thymidine. We then transfected the cells with NS1 and then lysed the cells with SDS, took the DNA and shared it mechanically just so it's easier to work with. And then we immunoprecipitated that with antibody to GFP, so that if there was any NS1 GFP attached to DNA, it would precipitate. And then we did autoradiography and Western blotting. And if you looked at the autoradiographs at the 100 kilodaltons point, where you expect to see NS1 GFP, we could see radioactivity. And the radioactivity is in the DNA. So presumably there was DNA attached to that GFP NS1, at least pieces of DNA. If you treated that lysate with DNase, you would reduce the amount of density of the autoradiograph bands, confirming that we were indeed pulling down DNA. And then if you then looked at a Western blot, you could, with antibody to GFP, you can show that indeed that 100 kilodalton band had GFP NS1. So the answer is yes, bulky adducts were formed. And so the model then for NS1-induced activation of ATR and PARP would suggest that NS1 forms a bulky adduct and breaks a single strand, which allows PARP to identify the single strand end and for RPA to identify the single strandedness in the near the bulky adduct. And as a result, these are energy depleting and it would lead to apoptosis. So what happens to that NS1 that has modified DNA during apoptosis? Well, we know that in apoptosis, cells end up condensing their chromatin and packaging them into apoptotic bodies, which are little blebs that come off of the cell membrane. And so nuclear material is condensed in those surface blebs and they bud off to generate subcellular particles. We also know that nucleosomes, fragments of DNA that have been protected because endonucleases chew up the DNA, but there are segments of DNA that are protected by histones representing about 146 nucleotides that end up in the milieu. And so we looked at, and this is very nice work that Leona did with her group, looked at scanning EMs of HepG2 cells and could show that in NS1 induced apoptosis, you see an increase in the amount of bleb shown as these little ridges on scanning EM. The D metal was a mutant NS1 that a metal coordination site had been removed and therefore it was not particularly functional. So you can see a decrease in the amount of blebs. So we were able to purify apoptotic bodies. Again, Leona's work, very nice work, in which we used, instead of the plasmid, which we had used in our original work, we used the baculovirus system that expressed the same EGF NS1. And we did that for greater transduction efficiency. And so we would transduce the HepG2 cells, add the insect steroid to turn it on, all the cells at the same time, and 48 hours later, collect the culture media, sticky fingers, media put it through a filter, a 5-micron filter, and collect, then centrifuge down and collect the apoptotic bodies, which interestingly would fluoresce green. If you look then under the microscope, and here's confocal microscope, you can show here's an apoptotic body. You can see it in under the Marsky microscopy. It would fluoresce green. It had EGF NS1 in it. It contained, you might actually see the red there, the red stain for the annexin, antibody to annexin, that showed that indeed the apoptotic body had phosphatidylserine on its surface and that it contained DNA by DAPI staining. And so this is merged, and you can see all three elements in that apoptotic body. And so what happens to the apoptotic body? Well, we also noticed that before we actually looked to see where it went, that these apoptotic bodies not only had EGFP and DNA, but they also had Smith and other nucleic acid associated proteins. And Smith protein is in lupus, antibodies to Smith is a very sensitive biomarker for systemic lupus erythematosus. So what happens to these apoptotic bodies when we put them onto THP1 cells as an example of an antigen presenting cell? These were phagocytosed. And here you can see a control with no apoptotic bodies. Here's an EGFP treated apoptotic body, sort of little GFP is taken up. There's really no apoptosis of bodies. But here you can see an apoptotic body with the NS1 and a lot of annexin on it, and ultimately demonstrating that indeed apoptotic bodies, or apobods as we call them for short, were taken up. So then we asked, what happens in a living organism when you see apoptotic bodies? And we looked at BALB/c mouse, a non-autoimmune mouse, and we exposed them to apoptotic bodies, which had been generated in HepG2 cells with expression of NS1. We also treated apoptotic bodies with staurosporine, which also induces apoptosis, and we also treated mice with pristane, which is known to cause a lupus-like disease, a chemical induction in mice. And then we sampled them 1, 4, and 8 weeks later. And indeed, we were able to show that double-stranded DNA antibodies were produced in these mice when they were stimulated with B19 NS1-induced epibods, and that the DNA, not only by ELISA, but also by Crithidia, which is a standard indirect immunofluorescence method that's used to detect double-stranded DNA antibodies in patients with lupus. And typically, these tend to be higher avidity antibodies because the Crithidia not only has a nucleus, but also has a kinetoplast, which is very tightly wound DNA, non-functional, but it's there and it's used as a marker. And indeed, we were able to show that these mice had generated a double-stranded DNA. What was also interesting is that we also were able to show that the mice developed encephalitis, myocarditis, hepatitis, and glomerulonephritis, manifestations that are seen in patients with lupus. And so in the brain, we could see demyelinization and degeneration of neurons in the heart. There were infiltrated immune cells and cardiomyocyte vacuolization. And the liver, we saw infiltrated immune cells and hepatocyte vacuolization and degeneration. And in the kidney, infiltrated immune cells and glomerular proliferation, hallmarks of systemic lupus erythematosus. And the severity scores, when you looked at how tissues are evaluated for severity of disease, we were able to show that indeed the NS1 induced severity. So what's the relationship between B19V and systemic lupus erythematosus? Well, we know that both of them share constitutional symptoms in common. They both can present with rash. Both will have non-erosive arthralgia and arthritis, and cytopenias and autoantibodies. And indeed, the question is, does B19 play a role in lupus? So B19 is common and widespread, and it persists. We have evidence that after an acute infection, it persists in bone marrow. And indeed, there's evidence that it will integrate into the genome of erythroid precursors. So the virus will stick around. And NS1 is typically suppressed in resting cells, but can be upregulated during cell cycle or under stress conditions. And so the question is, what would turn on B19 NS1 in a liver if someone had a remote infection? So thinking about the acute fulminant liver failures. So there may need to be a second hit. One of the hypotheses we have is that what turns on that NS1 is a second hit, which could very well be hypoxia. This virus likes to grow in erythroid precursors. The bone marrow oxygen tension is about 3%, and NS1 has an upstream regulatory element upstream of its promoter that's a hypoxic response element. So perhaps in hypoxia, NS1 gets turned on. And so we know that B19 can modify host cell DNA in non-permissive cells and induce apoptosis. There's some evidence that you can find B19 DNA by nested PCR in individuals with lupus, about 24% of lupus patients in this study. And we know that apoptotic bodies and nucleosomes are increased during lupus flares. So is B19 the only virus? It may very well be this is a good model, but it's not the only virus. And we know that Epstein-Barr virus has been implicated in some cases of lupus as a possible etiologic agent with cross-reactivity early Rho antibodies to EBNA1. Well, it turns out that what we have to ask, though, and we've asked John Harley, who first brought up the association of EBV and lupus, what is the original sin? What causes that cross-reactivity? And very likely, it's the fact that EBNA is also an SF3 helicase with structure similar to B19. So we proposed a model of SF3 helicase induction and acceleration of lupus, and that is this. So B19 will infect its target cells. NS1 will be produced and induce apoptosis, releasing nucleosomal DNA, in this case modified by NS1. Now, normally, if there is an anti-DNA B cell that is anergic, still in circulation, nothing happens when it takes up that DNA, even though it may have an anti-DNA receptor on its surface, an IgM antibody, surface antibody, because during ontogeny, the thymus has purged anti-DNA T cells. And so this B cell does not receive a second signal for help. But in this case, there are apoptotic bodies and perhaps even nucleosomal DNA that's modified by NS1 that's taken up by antigen-presenting cells, as we've shown you. The NS1 is processed to peptides and presented on MHC to the T cell receptor on anti-NS1 T cells that are persisted as memory cells since the initial infection. And that then is activated, and that can process peptide, present through help through its T cell receptor when the peptide that is taken up with the DNA by that B cell is processed and presented on its MHC as peptides, such that you now get a second signal that breaks tolerance, and that anti-DNA B cell goes on to become a plasma cell and make anti-DNA. So in summary, in terms of advancing the field, in terms of our scientific understanding, we better understand mechanisms of viral induction of autoimmunity and enumerating mechanisms of viral damage to host cell DNA. we hope that in clinical practice, physicians will appreciate that acute viral infection can present with features of autoimmunity and recognize that prolonged or persistent viral infection can account for chronic autoimmune-like symptoms. And the benefit to patients are improved diagnosis for perhaps differentiating B19 arthropathy from rheumatoid arthritis. The longest B19 arthropathy I've seen is 8 years. The patient never develops erosive disease. And they make 50% of acute adults with acute B19 arthropathy will look like rheumatoid arthritis and meet ACR criteria. And we'll treat them with drugs that they don't need and pat ourselves on the back and say, what a great job we did. We prevented erosions, but they were never going to get erosions. And the trick is, when someone presents with an acute onset arthritis, you have to think viral arthritis. You have to test for B19 IgM, because in 2-3 months that IgM may be gone and you'll miss the opportunity to make that diagnosis. And ultimately, one could argue that this would justify development of a B19 vaccine. Happy to take questions. And I should acknowledge the many people, some of whom, certainly in the University of Iowa, Yuri Karetny and Laura Cooling and Al Langness at the University of Nebraska took us in a direction of looking at livers. Brian Poole did some of the initial apoptosis work, and then Viola and Leona joined the lab and took this story further along. And when I left academia, I still got to play with very good friends with Viola, Leona, and Kai, who were at the University of Jyväskylä and continued the work. And the animal work was done at the University of Jyväskylä. So, and references for those who want to fall asleep at night with a good read. Happy to take questions.
And great, incredible, rigorous work. So much great presentation. This reminds me of the cases that we see in pediatrics, post-viral acute insulin-dependent diabetes, and immediate failure of their pancreatic cells. And then same model could be replicated to study more, also post-COVID. After COVID, we have increased cases of insulin-dependent diabetes, type 1 diabetes. So same model could be applied to study those. Do you have any feedback?
Well, it depends. If you want to look at those diseases, first you need a cell system that will support that viral growth, or at least expression of some of the proteins in that virus makes, and you need to be able to follow them. And that really becomes the first experimental hurdle in terms of identifying a system and where you can model that. So you would need to look, obviously, pancreatic islet cells. In this case, you have to be able to grow them in culture, infect them with virus, and see what is produced and what happens to those cells. That would be, I think, the approach that you would need to take.
Thank you, very talk.
Yes, welcome.
So correct me if I'm wrong, but SLE is more common in women. Do you think that this could be due to the fact that women are more likely to be in those high-risk jobs that you talked about? And is there any epidemiological evidence to kind of support and correlate with your data?
Well, we did not look at gender effects in our model system, but, clearly there are more women in those roles. Women are also tend to be child caregivers. And so perhaps there is more exposure, but there are multiple factors involved in regulating. So we express, for example, particularly with baculovirus transduction, we'll express NS1 perhaps in many more cells than undergo apoptosis. And so there are mechanisms in the cell to repair DNA. And so it may be a balance. And some people, their just genetics are a little bit better at repairing DNA. And so those individuals may be a little more protected from this kind of post-viral development of autoimmunity. So there are, again, multiple factors. I think the question of why women get certain autoimmune diseases more than men, and there are some men get more than women. I think that's still an open question. We like to think it's hormonal, but I think it's much more complicated than that.
So we have one last question from the honorary attendee, Dr. Tala. Are ANA antibodies seen after B19 infection in the absence of lupus symptoms?
DNA antibodies?
Are ANA antibodies seen
A-
after?
ANA. Yeah, many of the patients who get an acute B19 infection, even those who don't go on to, long-term, who essentially clear their symptoms within a short order, many of them will have transiently antibodies to rheumatoid factor or ANAs or double-stranded DNA. So we see that transiently in some individuals who don't go on to develop lupus. And, I think that when I was at the University of Iowa in particular, I saw any number of adults who had an acute onset of B19 infection. And many of them I followed who had persistent infection for some time. So none of them developed a lupus-like syndrome. So I think that this is background. This model is background for what might happen in people who do develop lupus. I mean, clearly John Harley's work went to the Department of Defense Serum Bank, looked at individuals who had been diagnosed with lupus and went back, the American Pentagon, now they're going to start calling it the War Department, started collecting serum annually from soldiers. And so he was able to go back years before and see that there were individuals who developed lupus had developed autoantibodies earlier with antibodies to starting with Rho. And what the antibodies recognized early on were epitopes that were seen in Ebna, suggesting that there may be a role. And then there was epitope spreading in the antibody. They would see antibodies to a lot of other things. So, yeah.
Thank you.
You're welcome.